when President Biden set up his administration almost 4 years ago, he writes from artificial intelligence to synthetic biology, and I don't know who wrote these words, but it's good for me. But the difference is, Stanford's had an artificial intelligence lab for about 60 years and a human-centered AI led by Fei-Fei Li for almost half a decade. And we're not organized in the same way. So these two things, AI and SB, are not the same, even though the president is putting them in the same sentence. So let me give you an on-ramp into biotechnology and how to think about synthetic biology, at least how I think about it. This is a photograph of what's thought to be a very advanced R&D facility from about 1,000 years ago. The terraced patterns in the land can be used to control irrigation and temperature, and obviously the angle with respect to the sun controls sunlight. And it's thought that in South America, this R&D facility was used to help accelerate domestication of the potato. And so the first generation of biotechnology is grounded in breeding and selection of plants and animals to have traits. And by doing that, you can adapt what nature provides in its bounty to feed an empire. So that's chapter 1 of biotechnology, the historical version, thousands plus years old. About 50 years ago, starting at Stanford, this is the patent owned by Stanford in 1980 on genetic engineering, all the way forward to today. Maybe you've heard about CRISPR. CRISPR is the 4th generation gene editing tool. This defines a second chapter of biotechnology, the editing chapter, where you can come in and make precise edits and change the traits of living systems more directly than through breeding. But I represent something still different, so we're going to have to get to that. And as powerful as CRISPR is, from my perspective, it's not that new. It's just more gene editing, which we've already been wrestling with and navigating for half a century. So let's talk about synthesis. This is a photograph of bottles in my office. These are bottles of chemicals. And I think of these bottles as ink cartridges for a printer. The bottle on the right, I just want to make sure, is my slide advancing okay? You can see the bottle, cool, thank you. The bottle on the right, if you look carefully, has the letter A on it. And then the next bottle to the left says T. And the second from the left says G. And the leftmost bottle, I didn't angle it right, has the letter C on it. So DNA is made up of 4 bases, A, T, C, G, and these bottles contain those bases in raw form. The chemicals in these bottles are not derived from a living system directly. It's just the individual bases. And you connect these bottles up to a machine, a DNA printer or a DNA synthesizer, and by pressing the keys on a keyboard controlling the plumbing, you can have these chemicals dispensed in whatever order you want. So the DNA printer or DNA synthesizer will make the DNA you want from scratch. So that's what I mean by synthesis, as an example of synthesis in biology. We can build DNA together from scratch, not starting from any natural material, not starting from any existing lineage or organism. Now, I'm hopelessly biased. I'll go out on a limb and argue that this is one of the most significant technologies of our time. The printing is still very immature. When I first started teaching 20 years ago, printing DNA cost about $4 a letter. But through innovation and translation into companies, we've gotten the price down about 100-fold. So today it costs about 4 cents a letter to press a DNA printing keyboard. I'm just going to move. Let me see if I can do this really quick so I can see the chat. Oh, great. Okay. So this is what I'm going to argue is the backbone of a 3rd chapter of biotechnology powered by synthetic biology. Let me try and give you a postcard to justify that claim. Back in 2020, when the SARS-CoV-2 pandemic was circulating around the world, you can track by looking at the public health records, when did it arrive in different locations? And so in the top left, I'm showing a photograph from the border of Italy and Switzerland. And we can ask the question, when did the pandemic naturally arrive to Switzerland? And the public health records say the 25th of February of 2020 is the first recorded case. But it turns out that's not how the virus first got to Switzerland. In January, a month before, a researcher in China had isolated the virus, used DNA reading tools to get access to the genome sequence, and put the information describing the genome on the internet. And the next day, a researcher outside the Swiss capital downloads that information and orders the DNA for synthesis, uh, putting in motion a process to rebuild the virus from scratch in Switzerland. And they managed to do all that and recover infectious virus in a laboratory by the 13th of February of 2020, or 12 days before the pandemic naturally arrived. So what just happened is you had atoms in China, they got decoded into bits, the internet moved them around to Switzerland, somebody remade the atoms encoding the biology, and now you've moved biology around faster than it travels in a pandemic. I would call this an example of a bionet transaction, which begs a question, what is the bionet? How would we build and secure the bionet? But it's powered by this ability to go from bits to atoms through synthesis of biology. This is qualitatively different than having an organism and editing it. In this case, you just have to have the information and the bioprinter to get to the organism. Now, remember, this is early times. The DNA printers and our ability to build biology in this way is super immature, right? It's like we're talking about AI at least a decade before ChatGPT. So freak out, but maybe don't totally freak out, right? It's not, we got a little bit of time to figure this out. So let's come back to synthesis. A lot of different ways to think about it. I like the ancient way of thinking about it. It's about putting things together, or composition, or a musical synthesizer, or a symphony. And so it just happens to be that I'm working at a time and our kids are learning at a time where synthesis is coming for biology, and it's up to us to sculpt and shape that if we wish. Here's a watercolor work of art from David Goodsell in Southern California, and this is representing the molecules of life. This is a depiction of part of the bacterium E. coli. And so on the left in green, you can see the flagellar motor, that is this pushing apparatus that spins around really fast and pushes the whole E. coli through. On the right in yellow, you can see DNA and RNA, the genome. In bioengineering today, when we think about synthesis, we mostly think about synthesis or putting together biology at this level, the level of molecules becoming cells. And we do this and are motivated in doing this for a variety of purposes. Let me give you an example from my wife's work. So I'm hopelessly biased, but it's a good example. So she used to be a professor at Stanford and then has left to run her startup company. What they did in the labs at Stanford was to take brewer's yeast or baker's yeast that you use to make bread or brew beer or wine. And instead of feeding that yeast glucose and having it make ethanol or bubbles in bread, they reprogrammed through synthetic biology the metabolism of this yeast to instead make chemicals found in plants. Chemicals, for example, found from the nightshade family of plants. In this example, they're making a chemical called scopolamine, which is used for motion sickness patches and for some treatments related to Parkinson's. But they can also make chemicals found in opium poppies, thebaine, for example. As they've taken this work out of Stanford and brought it into the lab of corporate labs and then commercialization, they're now running fermenters that are about the size of a swimming pool in Italy. And in 1 week, they can brew enough of a key starting material that would take 10 square miles of poppies grown for 2 years. In the examples demonstrated so far, they're making something called thebaine, which is the key starting material for the active ingredient in a Narcan inhaler or injector for treating an opioid overdose. So if you wondered, how do I get supply chain resilience or onshore manufacturing for essential medicines, synthetic biology unlocks full programmability of metabolism, and anything that you could reprogram yeast to brew, you could brew wherever you want to do the brewing. To give you an example of how quickly this goes from fundamental ideas around how do we put things together in DNA synthesis to something that might impact ourselves or people we care dearly about. Let me give you a different example, also coming out of Stanford. Phil Ross earned his Master of Fine Arts in Stanford. He got very excited about working with mushrooms or wood fungus. And one of the things he and his colleagues learned was if you grow a wood fungus on a lasagna tray of wood chips, if you can imagine that, or a lasagna tray of sawdust, over a period of time, the surface of that tray will differentiate into a type of mushroom skin that you can peel off and put through a tanning process. And so this has become a company with its R&D facility in Emeryville in the East Bay called MycoWorks. They've just moved their headquarters to Paris to be near the fashion houses that are incorporating this into luxury goods. But the factory, their first factory, is set up in Union County, South Carolina, and it's just been coming online over the last 6 months or so. And so what this factory does is it takes sawdust from the lumber mills in South Carolina, feeds it to the wood fungus, and they can grow about 6 million square feet of leather surrogate or mushroom leather a year. I estimate that's about as much leather as you would get from 60 to 80,000 cattle a year. And if it was cattle, they'd be grazing on 200 to 300 square miles. But I'm not an expert in cattle grazing, so maybe somebody can figure that out and double check my math. But what does it mean when we do synthesis with biology? And suddenly the examples might surprise you. Here's another example. Oftentimes we think about food and medicine and materials. The Defense Department and the Navy back in 2011 are asking, could we do brewing of energetics? And energetics is a fancy word for high energy compounds that you could use as solid fuel for a rocket or a missile. And suddenly, like anything we can encode in DNA could be grown anywhere increasingly. All right, so that's what's hopefully giving you a taste of what's up for grabs. Our children are aware that biology is important and that it's becoming a general purpose technology. This is a program I helped start 20 years ago at MIT. It's now called iGEM. It's a genetic engineering Olympics, like FIRST Robotics, but it's for bioengineers. We have about 5,000 students a year participate. Stanford has an amazing team. And the end of competition jamboree is in Paris. President Macron's office helped provide cover for this. And so we see this next generation of people wishing to explore and embrace and engage with biology as a platform for realizing a flourishing future. With all this happening, it's still early, and I think we don't quite understand what biology could be as a technology. So with this metaphor of a leaping frog, how to think about this. One thing I think about is how caught up we are in the practice and rhetoric of industrialization, concentrated capital, concentrated energy, concentrated factories, and how this is the opposite of what biology does in nature. The leaves on a tree don't wait for a CEO to say start growing or in a presidential executive order. They just grow with the energy arrives and the materials that arrive where the tree is going to be, right? Biology's superpower, if we think about it, for me, is that all atoms are local. That's what biology teaches us. And what synthetic biology does is it allows the superpower of the internet, moving information around, to potentially converge with the superpower biology. So we could design DNA anywhere, move it around, and grow biology so programmed wherever. And what's interesting is everybody everywhere's got biology. And the industrialized economies and the centralized supply chains really only provision to about 2 billion people. Most people don't have reliable access to World Health Essential Medicine list. And so what would it mean if we had a distributed manufacturing platform connected to the internet, or deglobalization in certain ways? This is a photograph I took on the outskirts of Guadalajara, Mexico. And you can see the 18-wheelers on the dirt road. But here's the biotech company. This biotech company is shipping about 30 million foil packets a year into North America and Europe in the probiotic aisles of the supermarket. So maybe they're doing retail pricing 20 to 30 million revenue a year. What happens when they have access to higher value biocontent through a bionet? Another thing that's very interesting is, even though PG&E in California keeps raising our rates, our ability to generate electricity seems to be on a physical basis, trending towards a cost of zero. And that means we're transitioning to electricity generation abundance for the first time in human history. We think about the challenges of storing energy in batteries when we have surplus energy production. But another thing you can do is you can use that electricity to fix carbon out of the air and make a surrogate for glucose. So instead of having yeast grow on glucose that comes from sugarcane or a sugar beet, you could have yeast grow on carbon that's fixed from the air using photovoltaics. So we need a better word for this, but right now we're calling it electrobiosynthesis. And this is a complement to traditional agriculture. Anywhere you could generate electricity, potentially you could grow biomass, right? And all the PhD students showing up in bioengineering, about maybe 20 to 25% of them are starting to go into this area of research, which suggests it probably is important. Now, I put these sorts of things together and extrapolate, which is risky. and think about what happened 50 years ago across the creek in Menlo Park from campus. The computer became personal back then. And the revolution wasn't in transistor physics or even too much in the electrical engineering details. It was in the terms and conditions of who could compute where for what purpose. And so biotechnology right now is locked up and industrialized, and it's not that personal. Although sometimes we talk about personalized medicine, it's not really in the terms and conditions of who can do biotechnology where for what purpose. But if I can get energy and use that energy to get carbon and nitrogen, and I can bring biocode in, then I could have a personal biomaker. And maybe it's not mine, maybe it's my family's or my neighborhoods or my towns or my district in the US House of Representatives, but maybe it is. I still remember when my mom brought home a Franklin Ace computer in the 1980s, the early Apple clone before it was sued out of existence, and how important that was for me as a student, as a learner. And so just to provoke and think, and remember, this is the emerging third chapter of biotech, synthetic biology, and we're finding our way. And one of the things I found to be important to say, especially in policymaker audiences, like, what if I'm right? This stuff's so important. We need strategies that are not just the conventional strategy, but hedges around how this might go. I want to share a little bit about the fundamental research we do here on campus. This is a project we're just starting to publish. The graduate students have just earned their PhDs. This is our work to build a cell from scratch, to construct life from scratch. And what I'm showing here is a electron micrograph. It's a fancy microscope up at the National Lab. It costs about $10 million. And the image here is a false color image of this thing we've built, which is a synthetic egg, if you will. Every molecule in this object is a purified molecule isolated from one of 6 or 8 different natural living organisms, but then reconstituted by hand in a test tube to form this lipid-enclosed membrane that's got molecules inside it that can express any DNA we add to this system. And we're using the microscope just to see where the molecules are and if it looks right. And when I get asked questions about this, people will say, well, what egg did you start with to get rid of the... No. This is not derived from an intact organism. This is literally Humpty Dumpty, if you know that nursery rhyme. But imagine you had eggs from 10 or so different organisms and you just picked up the pieces and tried to get one thing back together. Another sort of question I get is, well, is this like E. coli, a bacterium, or is it like yeast? What type of cell is it that you're building? And I say, no, it's an American cell. It's representing a set of political values around who can work with it in cooperation and community, sort of like Unix at Bell Labs decades ago being developed to support a fellowship of software programmers. Anyway, this cell by itself is not yet alive. For that to be true, we need to figure out what DNA to add to it to get it to grow and divide. And if we can solve that problem, and as we solve that problem, on the other side of that, then you'd hear an announcement coming out from here or wherever it happens first saying, researchers, bioengineers have constructed a simple life form entirely from scratch. The significance of this is not that achievement so much as when we work as bioengineers today, there is no natural cell on Earth that's fully understood. And so anytime we do a bioengineering project, it's very Edisonian, it's very tinker and test in our workflow. Imagine the building you're in right now, or the next car you go for a ride in. That building you're in hopefully wasn't a research project that required Stanford postdocs to get the building to work. Instead, it was just reliable, mature engineering with good mathematical representation of how things behave. So it's not tinker and test, it's design, build, work. So whoever can get to the operating system for life at the cellular scale will realize a foundational breakthrough for all of biotechnology that makes it, in my view, a much more mature, much more distributable, much more reliable platform for making things by growing. And it's hard to think about this. So I wanted to share a poem and a photograph of my maternal great-grandfather, Parker Wilson, excuse me. He lived in Washington County in Western Pennsylvania and played the fiddle. And if you know the poem by Hardy, I am the family face, flesh perishes I live on, projecting trade and trace through time to times anon, and leaping from place to place over oblivion. Like, I look a little bit like him, and I had nothing to say about that. It's just our facial morphology has some shared lineage, literally. All life on Earth naturally is constrained by lineage. We derive from the life that comes before us. And it's also constrained by this physical requirement of needing to be able to reproduce through direct descent. Our boys, Christine and I have two boys, Sam and Max, and that was not easy. And then the other thing is life is constrained because the planet we're on changes. And so over time, life has learned how to support evolution. It's evolved to evolve. And so there's 3 layers of constraints on all the living systems that exist on our planet right now. And just like in physics, we might be familiar with how mass creates a distortion in space-time that results in a gravity well, and you need to rocket up and away to get out of the Earth's gravity well. I also now see the Earth as containing a life well. All the life on Earth is at the bottom of the Earth's life well, constrained by these requirements of lineage, reproduction, and evolution. And what synthesis represents is climbing and crawling our way up to the lip of the Earth's life well. And I find this much more interesting than getting to the vacuum of space. Instead, we're just now, if we can get there, poised on the possibility of anything life could be, which is both enthralling and terrifying, depending on your view of the human condition. So let me close and transition into the final act of my remarks and hope to get to good discussion. Biology really could become a general purpose technology. It's not only about making medicines and food and stuff like that and energetics for the Defense Department. Like here, for example, speaking from Silicon Valley, is the Semiconductor Research Corporation's roadmap for synthetic biology. And what the heck's going on here? Well, the gist of it is, if you did 20 years of fundamental R&D, it might be possible to be able to eventually do bottom-up self-assembly of computer hardware, where you're positioning the dopant atoms and other things exactly where they need to be. The resulting computer is not alive, but you have a biotic fabrication process instead of a top-down lithography process that has been so dominant for the latter half of the 20th and early 21st century. What I like about this is it's physically imaginable and practically impossible today. Like bioengineers of 2024 have no chance of growing computers as imagined here. But if we did the fundamental R&D over the next 2 decades, and that might cost us, wait for it, about $100 billion, so $5 billion a year, we could probably pull it off. And in so doing, we'd unlock biology as a general purpose technology for growing anything that biology could grow. So I like this as a star in the sky, motivating discussions about how do we sustain investments in fundamental engineering research, which as yet really has been missing from bioengineering. The world I encounter mostly doesn't like change and doesn't like what I have to say. Senator Warner of Virginia, Mark, who I respect and work well with, sometimes will say I'm the scariest person he knows, in part because I represent change. And I wrestle with this and wanted to bring a history book in. This is a photograph of what I understand to be soldiers in the Maginot Line serving in the French army in the 1930s. And they're worried about vitamin D deficiency. And so they're being very rational and exposing their skin to UV light so they don't have to worry about vitamin D deficiency. But wow, did they have bigger problems? And they have bigger problems, as Marc Bloch, the Frenchman and historian, documents in his book, Strange Defeat, because people 100 years ago in the 1920s in France struggled to figure out how to navigate what was changing and to lead into the future. And, Mark writes and details what went wrong. Coming out of the last war and the 1918 flu pandemic, just want to get back to normal. And we were really tired. And so we just came out of trench warfare and we built the best trench we could called Maginot. But it doesn't work. And his language is much more powerful than my own. I'd encourage, it's a small book, but it weighs very heavy on me. I'd encourage anybody to check this book out. I'm very nervous that what I represent is structural change at a root technical level, impacting almost all of civilization, and we're managing it for status quo right now. So would welcome colleagues trying to navigate these types of discussions. To wrap this up, I want to just touch upon a couple points. Remember, the personal computer was a revolution in the terms and conditions of computing. So when I think about emerging biotechnologies, I think about how it manifests in the broader world, in America and globally, and what the terms and conditions will be. And so I want to highlight, there's several examples here, and Pradeep has a PDF copy of my slides, and feel free to ask him for a free copy if you want to double click on any of the links in here. But I want to highlight on this slide the work of Alondra Nelson from Princeton, where she has studied the Black Panther Party and asked questions. Why were they running public health clinics in Oakland decades ago, doing things like providing testing for whether or not somebody was carrying sickle cell genetically? And the answer to that, in part, includes a consideration around whether or not, if you went to the doctor, you would risk harm depending on your skin color. And to avoid that oppression, it becomes important for people to have the option of doing something differently. Stated in a different way, whether it's Tom Jefferson on the top left or the Black Panthers on the right side of this, there's a political theoretic argument to be made that if there's something that is essential for your livelihood or living or existence, and there's only one way to get it, whoever has control over the one way to get it could potentially oppress you. And so at a root level, we need to think about implementing optionality around access to essential capacities in order to guarantee a future in which people can be citizens of the future, as opposed to consumers or subjects or objects. And for my whole life, I've seen, in the United States at least, a struggle to have these conversations, but it's interesting to go back in time and see when we got it right, 200 years ago and 100 years ago, for example, with rural electrification. I think we desperately need to renew this type of conversation for all emerging technologies, but especially biotech. In this spirit, there's a postdoc, a Stanford grad now working with us, Callie Chappell, and she's inspired by the work of the Carnegie Endowment from the 1890s to the late 1920s around literacy and citizenship, and how the Carnegie Foundation helped build over 2,000 public libraries. Here's one I showed to Senator Todd Young when I was meeting with them in Washington week before last, from his hometown. And so what if we replace the first letter I in library with the letter A, and now you get a library. And what if we created a new profession called the librarian? And what if we got 2,000 public libraries all throughout the United States this decade, so that any American could become a citizen of the biotic future and help figure out what they want, right? Another thing to flag is how terrifying some of this is. And I learned this from an art professor, Paul DeMarinis here at Stanford. He said he was taking Introduction to Bioengineering one year, and he wrote me a note. He's like, "You're not talking about how scary it is, what you're doing." I'm like, "What are you talking about?" He's like, "Your bioengineering stuff's scary." And I didn't understand what he meant until he said instinctive fear. And then I understood what he meant. So my wife, Christina, is instinctively afraid of spiders, top left, or I am instinctively afraid of falling from heights. And I don't know why that is. I could tell you some story, but I honestly don't know. It's just instinctive fear. And so what if biophobia is real? What if there's instinctive fear of biotechnology? I believe it's real. And the issue is we haven't acknowledged that. Instead, what we've done is we've said, we're going to put the biotechnology away in labs, so it'll be biosafe, and that mostly works until it doesn't. And then we're going to tell you it's useful. We're going to make some utilitarian argument that it's useful. But the Nobel laureates have really failed to understand this instinctive fear of biotechnology. And fear's gift turns out to be the opportunity to become collectively courageous. When I admit that I'm afraid of heights, I don't get laughed at too much. Instead, we develop a profession around it so that I will drive across the Golden Gate Bridge, even though I can imagine it falling down and we're all dead. But I'm courageous because we're collectively not laughing at each other and figuring out how to make something possible, right? Both psychologically possible and technically possible. Part of the answer to this, in terms of how to get there, is revealed in the work of the Dutch social scientist from 90 years ago, Huizinga, who talks about play and how it's important for play to predate culture and utility. So puppies playing, they're learning how to bite without hurting each other, right? And in biotechnology, for the most part, we've skipped over the play phase because it's been too serious. So just want to leave a post note there in case anybody wants to think about that. Here's where I grew up and went to school in Bethlehem, Pennsylvania, the Lehigh River and Bethlehem Steel. And I used to hop trains on these rail lines. I don't recommend that. But now what I see is I see a fresh water supply, I see infrastructure and flat land. And I think about that factory that MicroWorks set up in Union County, South Carolina. And I think about how we might unlock $1 trillion of capital to do in the United States domestic biomanufacturing and creating a biobelt, right, underwritten by biobonds or something like that. The examples of the manufacturing content that would make viable businesses are just starting to surface, and it's very tricky to bring it to market, but that's a job we've got in front of us for the next 2 decades. So to close, you know, I'm a long ago civil engineer before I'm a bioengineer. Here's my favorite example of biocivil engineering. This is a suspension bridge grown from the roots of a rubber plant in India. And this bridge fixes carbon as it gets stronger. It's passed down from one generation to the next. It reminds me that nature is powered by photosynthesis, and that's 100 terawatts of energy, 5 times more than what our civilization runs on. The napkin math suggests that the physics of flourishing is very good. If we could partner with the living world correctly, it's seemingly possible to enable 10 billion people to flourish as earthlings, along with the rest of life on Earth, without trashing the place. And if we did it right, not only would we get to planetary scale flourishing, we could embed, you know, the politics of democracy, lowercase liberal democracy, in these capacities, and off we'd go. And I'd like to see us get this done and dusted by or before the year 2050, literally within a generation. And obviously that's not the work of one academic. But so just to, you know, like the stakes are high. Here on campus, we're organized in a bottom-up way. And so, you know, you could go to ai.stanford.edu and learn about artificial intelligence. If you go to sb.stanford.edu, you can see what we're up to. Thanks for your patience with me pushing these postcards, and happy to talk about whatever's on your mind.
Thank you, Drew. That was fascinating to us, you know, talk on a subject that I think most of us know very little about. So please chat in your questions for Professor Endy that you might have. But let me start off with one. So from synthetic leather to food to power to medicines, it looks like synthetic biology can cover almost anything. What do you think in the next, say, 10 years, what do you think might be some of the things that we might actually see happen?
Well, you know, if you go to a website for a company called Light Bio, starting in April, they're shipping bioluminescent to petunias. So bioengineers have reprogrammed petunia plants to emit light. All plants are powered by light naturally, so they harvest light and make sugar. These plants do that, but they also convert chemical energy into light energy. We had the early versions of these plants maybe 3 years ago, and my kids who go to school in the Palo Alto School District came back from school one day and told Christina and me that GMOs were evil. And as parents, Christina and I were just looked at each other, caught each other's eye, and say, "Don't say anything. You know, just be quiet." And then it got worse, Pradeep, because they saw the latest Jurassic World movie, where the evil multinational corporation is called BioSyn, literally a transposition of SynBio, what I do. And the CEO looks like Tim Cook and I somehow made a chimeric offspring. It's 90% Tim and 10% me. And it's like, it got even worse. But when I brought these bioluminescent petunias home for winter break and their cousins were visiting, they went into the bathroom and turned the light off and shut the door and let their eyes adapt to the dark. And they came out a couple minutes later and they said, "Cool." Right. So in the short term, like, check out bioluminescent petunias if you want. They're not super bright. It needs to be dark, but you could use them as night lights that you water instead of plug in. You know, if Christina's company works, you know, they're probably delivering 50 to 100 essential medicines globally, you know, they're actually starting this year to supply, right? But when you get up to 50 to 100, that's by the end of the decade. I think MicroWorks is going to work. You know, it's like finding your way in an emerging frontier means there's a lot of failure, right? So in my niche, it's, you know, when I talk with Feifei on AI, she's worried about Microsoft and whether or not only big corporations like Microsoft have the compute power for AI model training. And I say to her, like, wow, I'd like to have that problem because we don't have Microsoft, right? There is no like huge company making huge amounts of money in my space yet. So there's going to be innovations, and the early innovations are basically changes in supply chains that initially don't look like totally different products, except for maybe glowing plants. It's just we're making something in a more sustainable, resilient, distributed way. But once those capacities get made, then it's not just a replacement for animal leather, it's a better material that maybe has some sensing and healing properties or does something totally different. And that'll take a little while longer. Another thing I am seeing from the undergraduates, first of all, and now it's getting into the labs, is fully biological technology. Most biotech is hidden away in tanks and fermenters. But students on the Stanford iGEM team in 2020, their project was in the middle of a pandemic. They were so frustrated by how hard it was to learn if they were infected. They decided to do something about it. And so their project idea was to re-engineer a bacteria that's a friendly bacteria that lives in our sinuses to naturally uptake genetic material that might be in your sinuses and then determine what that material is. And depending on what the material is, they would change the color of your mucus, is their idea, right? So if you were infected by an influenza strain, maybe your mucus would be bright pink. And if you were infected by a coronavirus strain, your mucus would be bright purple. So to my kids, I explained their project as if you'll forgive me, the Purple Booger Project, right? And instead of you send a sample out to be tested, you have a living, pervasive surveillance platform in your nose. And so this is an example of fully biological technology. More recently, Michael Fischbach, a colleague in bioengineering, his team, there's microbes that live on our skin, friendly microbes that live on the skin, Staph epidermidis, for example, and his team reprogrammed it to present an antigen to train your immune system to attack skin cancer. So could you have a living skin cream that vaccinates you against melanoma? And who wouldn't want that? And of course, there's the challenge of cancer has been cured in mice thousands of times. Who wouldn't want to be a mouse? And will that work in people? We don't know yet. But I do think a different category of bioproducts, which will just be fully living, distributable bioproducts. Yeah.
Okay, it sounds like there's so much potential here. So one question is, what would accelerate your growth 10x if you were to say these were the things we would need? What would you?
It's a great question. In general, the United States over the last 50 years has drifted away from supporting foundational research, even though we do support some. We tend to support more and more translational research. What is this technology good for now? And it's even more acute in foundational engineering research. So there's good support for foundational science, but foundational engineering research is almost impossible now to fund. And it didn't used to be this way. We used to have strategic investments in computing and networking and so on. And starting in the 1980s and 1990s, we just drifted away from that. So the National Science Foundation, for example, through the CHIPS Act, has a new directorate that's richly funded called the TIP Directorate. And I'm not arguing against it, but the T in TIP stands for translation, right? So I just want to point out that in general, we've been hollowing out our investment in foundational engineering research. And then it's even worse in biotechnology, because biotechnology is a technology of last resort. And so the teams in biotechnology tend to be overdriven by immediate utility. It's like cure cancer, please now, right? It's a valid request. And if you could cure cancer by going to Ace Hardware and getting nuts and bolts and doing it, you do that, but you can't. And so you come to the bioengineers and it's the doorstep of last resort. And so people in my world tend to be overdriven by the immediate pressing need. And again, that's not bad, but it creates a overweighting towards immediate application and an underinvestment in the foundations. It would be as if I was a computer science professor and my students could only do projects that were make a mobile phone app that helps a patient in a doctor's office tomorrow. And if I said, I want to work on a new operating system or I want to work on a new programming language, my dad would ask me, like, what disease is that curing? And the answer is a little bit meta. It's curing the disease that diseases are hard to cure, right? And so in general, the answer is, please, for the love of all that's good, could we sustain foundational engineering investments? What does this look like? It might look like building national labs for the 21st century. Like the 20th century got labs for the physicists. Teller and Oppenheimer actually got their labs. And we have the legacy maintenance of those labs, including here on Stanford's campus. And I've not been able to work through Department of Energy to refactor any of that. it's interesting to see China going all of nation on synthetic biology and biotechnology. And I can show you what national labs look like because they're building them in Shenzhen. They're really cool. And we've got nothing like it anywhere else in the world. So that's part of it. More investment in the foundational engineering, more investment in the tools layers. There's so much student interest and demand for all of this, but we just keep struggling to do the low-level innovation. Another thing is like we got to make biotechnology cool, and that's not the only way to describe it. But if biophobia dominates always, and then people are consumers mostly through medicine, but don't have standing or literacy, right? That's not a culture that's a flourishing biotic civilization. You know, that's something very pathological, right? So unlocking biology as a general purpose technology by organizing capital to do the sustained tooling investments, and then working on what it means to have a biotic culture, a society. Both those things are very much on our mind. An idea we have around this is, you know, some generation ago plus, Xerox PARC was an interesting research lab that pushed what was happening in electronics and networking and how we might interact with computers. And so we're thinking about doing something, we'd like to do something like that, but for biology, biotechnology. So we're calling it Biotic on the drafting board right now.
Okay. So there's a question, is there a master's degree that students can earn working on this type of area, and what's the name of that program?
So in the bioengineering department, we have an undergraduate degree, a bachelor's degree, we have a co-terminal master's degree, and we have a PhD. So it might be the co-terminal master's degree would be of interest. Another thing I should have mentioned is Professor Brophy and I, we teach Introduction to Bioengineering, and all our course materials are free online and are on YouTube. And so if you'd like, as follow-up to this, or somebody you know might be interested, feel free. I just dropped the URL in the chat, introbioe.stanford.edu. And we don't teach from a book, so it's always work in progress, and we're happy to receive feedback. The other thing that's interesting about this course is we have interviews with people who are not biotechnologists, asking them what type of bioengineer they are. So for example, the actor Ahmed Best, who played Jar Jar for Lucas, is a colleague and friend, and you know, we ask him what type of bioengineer is he, right? And so there's interesting things on that website.
Okay. And can you explain in layman's terms how someone can take the digital code for something and make it in the lab? You like,
Yeah.
for example, you described the COVID BioNet before it arrived.
Right. So imagine you have a 3D printer that you or your kid is using to print a plastic bunny rabbit, right? And it's dispensing some polymer on an XY platform that builds up the rabbit, right? And how is that done? Well, you have a computer file that instructs the printer how to dispense the plastic. The bioprinter is even simpler. It's a 1D printer, and it's just printing a strand of DNA by dispensing the chemicals in a particular order coming out of those bottles I showed you. And however you program that printer, you'll get the A's, T's, C's, and G's in that order. So it's just a 1D printer. But here's where it gets amazing because it's biology. You put that 1D molecule of DNA, that string of DNA, into a cell, and now that's the code that the cell runs on. And of course, the cell makes four-dimensional objects, space and time, developmental biology. So at the heart of synthetic biology is a 1D printer, the DNA printer, and then that DNA program controls a four-dimensional object, living matter. So in the abstract, that's what's going on. Anything you wanted to, so here are some things I would wish for. Remember how difficult it was to get supplies of the vaccines? And we're always wondering about vaccine equity and who can get it. And, you know, like right now I'm on a study for the Strategic National Stockpile on smallpox. And, you know, most of the United States is not vaccinated against smallpox anymore. And so we're worried if there's a smallpox outbreak, how are we going to get everybody the vaccine? So we have a strategic national, just like we have a strategic oil reserve or strategic cheese. I don't know all the strategic reserves we got, but we've got a strategic reserve of vaccines. What if instead I could just download and grow a vaccine in my kitchen, right? And do that safely and reliably. Would you imagine that people would worry about Wi-Fi and Bluetooth chips in our vaccines if they were all homemade? and it was like a nice homemade meal, like a Thanksgiving meal. And it was the good stuff because you made it out of love for your family, right? So imagine you had a distributed capacity to manufacture diagnostics, treatments, vaccines, uh, vitamins, right? That's why I'm so intrigued by this idea of a bionet and pushing the limits of decentralization and some aspects of deglobalization.
We're running out of time, but this is an interesting ethical question, which we'll take this as the last one. It says this potential is both exciting and terrifying. So we appreciate there are thoughtful people like you and who are considering the issues, but is there a danger of unintended consequences and power in dangerous hands?
100%. It's why I let me double click and foot stomp that book, Strange Defeat, and how hard it was 100 years ago in France, at a point in time where there were a lot of other challenges and everybody just wanted to get back to normal. And the technical innovations of the day were the internal combustion engine and the radio, right? And some could see that maybe troops in the army would move around differently given tractors with internal combustion engines and radios, right? But nevertheless, they still built Maginot. There are very significant challenges ahead of us with respect to safety and security. There are very significant challenges ahead of us in terms of, in the United States, I'll just use the United States, for example. I know it's a broader audience. Like President Biden in September 22 signed an executive order on America's bioeconomy. So what's going to make the bioeconomy American? Is it that the jobs and money are in the 50 states, or is it about the terms and conditions? Is biotechnology only about life, food, fuel, medicine, or is it also about liberty and the pursuit of happiness? And I don't mean that as just words. I mean that in terms of we've got to get the politics and culture of it right, in addition to the safety and security of it. And all of this is up for grabs right now, and it mostly does scare policymakers. And we're at risk of making mistakes because of that, right? So let me give you a live example, and I know we're at time, but let me still try and land this. The more recent executive order on artificial intelligence in the United States has a whole bunch of stuff on synthetic biology, which I didn't expect to be in there. And one of the things it says is, if you receive federal funding and order DNA from a DNA printer, you have to have what you're getting printed checked through some central checking process. Only by doing that, and the reason is for biosecurity purposes, so that somebody isn't getting access to some pathogen, right? And so it's well intended from a security perspective. But if you think over the longer term, we're talking about governance of a printing press. And so how should we really go about it, right? And it's all just up for grabs right now. So I don't want to be, I don't think there's, well, I should be careful. I think we're in a moment where there's significant ambiguity around the ramifications of what's emerging in biotechnology, and it impacts all of us. Each of us has the option of digging into this and learning more and becoming a protagonist within it. But it's a real live topic, and you shouldn't come away from this conversation feeling settled. Like things are up for grabs, and it's a big deal.
Thank you so much, Drew, for your talk today. It was a fascinating subject, and I think I think we're going to see a lot more of it as we go forward. So thank you for that. And by the way, you'll be pleased to know that you just earned yourself 2 bottles of our priceless Stanford Parents Club jam. But thank you so much for your time today, and really appreciate you coming and talking to our parents.
Thanks, Pradeep, and everybody. I'm grateful to be connected with each of you. If anybody would like to follow up, my email is just my last name @Stanford, and I'll do my best to be in touch or point you to whatever resource might be of interest.